Hello everybody, today we're going to talk about the process of protein synthesis. What does synthesis mean? It means to make something. So the process that we're going to illustrate today is about how your body makes proteins. It has two main parts. The first part is called transcription. Notice it has a C in it. This is going to help us remember the order because following transcription is going to be another process called translation. And you'll notice that they are in alphabetical order. C happens first and then translation. So we're going to just start right now with talking about the process of transcription. So our bodies have DNA in them. And during the cell cycle, the G1 of the cell cycle is when the cell is busy making proteins. Okay, so this happens during G1. What's going to happen is that the DNA is going to split apart at the region of the DNA where a gene is that needs to be made into a protein. So the enzyme helicase is going to come and unzip the DNA, and then similar to when we were making new DNA, there's going to be an enzyme called polymerase that's going to come in and build on, hammer, I'm building on new nitrogen bases. In fact, entire new nucleotides are being assembled. Okay. We only copy one side of the DNA, and this process that we just did is called transcription. Transcription is basically going from DNA to a molecule called mRNA. M is for messenger, because we have to somehow get this information that is stored in the DNA out to the ribosome, where we had learned earlier in the year that ribosomes are where protein is made. So this right now is happening inside the nucleus of the cell. So this is the nucleus. When is this happening? It's happening during G1 of the cell cycle, which is part of interphase. So we already knew from before that DNA cannot leave the nucleus. So we have to take the message in the DNA and put it in a format that can leave the nucleus and take the message to this organelle called a ribosome. So that's what messenger RNA does, is it's going to take this message and it's going to remove it out of the nucleus and take that information to the ribosome where it can be read. Okay, so messenger RNA delivers the message. mRNA delivers the message. Where is it delivering it to? The ribosome. It's taking the message that was amongst all the other DNA in the nucleus. A copy of just a gene was made, and that message is sent to the ribosome. At the ribosome, we are going to read 3 nitrogen bases at one time. We call those 3 nitrogen bases, so right here, I'm going to label it. These 3 nitrogen bases together we call a codon. and we're going to read 3 at a time. You will notice that my ribosome, I made it so that there's a window in it, and that window is just large enough for a codon. And what's going to happen is that the mRNA is going to slide along the ribosome, and we're going to read one codon, then the next codon, and then it's going to stop at the next set of 3 nitrogen bases, and then it's going to go to the last set of nitrogen bases called a codon. So a codon is a set of 3, 1, 2, 3 nitrogen bases. So again, there's a codon right there. So how does the process of translation actually occur? The messenger RNA is going to be read at the ribosome 3 nitrogen bases at a time, called a codon. See, I labeled these 3 down here a codon. As it is read, another molecule is going to come and try to pair with it. That molecule is called tRNA. T for transfer. T is also for its shape. Can you see it's shaped a little bit like the letter T? This is a cartoon version, but if you were to look at the shape of the actual molecule, you would see that it is somewhat shaped like the letter T. So the tRNA has also 3 nitrogen bases on it. Those 3 nitrogen bases are called an anticodon. The codon and the anticodon will have to match in order for the amino acid that is being carried by the transfer RNA to be dropped off. So this here is representing an amino acid. That's this acid. Okay. So let's see how that process is going to work. So first step, we have our window of our ribosome over top of one codon. It reads AUG. So we need to look, does this tRNA match? No, it does not. So this is not the right amino acid to attach. Does this tRNA match? Yes, it does. Uracil pairs with adenine, adenine pairs with uracil, cytosine pairs with guanine. So they match. The tRNA is going to drop off its amino acid, and then the mRNA is going to slide down, and we're going to read a new codon, a new set of 3 nitrogen bases. What would pair with AUU? Well, a tRNA with an anticodon of UAA would pair. So click, they pair, and now the amino acid is dropped off, and we now have so far a polypeptide chain of 2 amino acids. These tRNAs go out back into the cytoplasm where they can pick up another amino acid. The mRNA now is going to slide down, and we're going to read a new codon of 3 nitrogen bases, GUA. Well, which amino acid will pair? Which anticodon on the tRNA will pair? This one will. C pairs with G, A pairs with U, and U pairs with A. So this amino acid can be dropped off by the tRNA molecule. Finally, we slide to the last codon. We find the matching anticodon and drop off the last amino acid. So we now have a chain of amino acids that we call that a polypeptide. polypeptide. Now you may say, hey, I thought a bunch of amino acids attached together was called a protein. Well, you're close. It's first called a polypeptide. After the polypeptide is folded and twisted and made into just the right shape and attached to some other polypeptides, then we will have what is called a protein. So to summarize, protein synthesis is made up of two separate steps. The first step is called transcription, as a C, so it goes first. The second part is called translation, where we are actually changing the message into a very, very different thing. So transcription is when we are simply changing the message to a slightly different format called messenger RNA, and then we're going to send the message out of the nucleus. Translation, we're taking this message, which was made out of nucleotides, and we're changing it to a very, very different format. We're translating it. Here we read the message 3 nitrogen bases at a time. So we read it 1 codon at a time. We paired the anticodon on the tRNA with the codons on the mRNAs. Each time we were able to drop off 1 amino acid, resulting in, just set that back there, resulting in a polypeptide. Huh? I thought we were making proteins. Well, once this polypeptide is folded, we have a protein. So once polypeptide is folded and attached to other polypeptides, then we make a protein. So I'm off the screen. Other polypeptides, we make a protein. There we go. So once the polypeptide is folded and attached to other polypeptides, we call it a protein. You may ask yourself, why do we call it a polypeptide? Well, because the name of the bond between these amino acids is called the peptide bond. There's many peptide bonds when we attach amino acids together. And once it's folded and twisted and attached to other things, we call it a protein. So this whole process is about making proteins. Your body does this all the time. This is happening during interphase of the cell cycle, and it is going to be responsible for how we look and how our traits are displayed by our body. We'll talk about that in another video.